Good morning. You'll notice I've added a few qualifiers to my talk title. There's no surprises today. I'll be talking about rats as usual. So I thought before I settled into my comfort zone, I should briefly acknowledge that there are a lot of different isotopes and a lot of different ways that specialists in isotope analysis are pushing the boundaries of what we can understand isotopically and about the past. So paleoclimate, migrations with strontium to varying degrees of success in the Pacific, and compound-specific approaches are an especially exciting new method that can really refine our understandings of diet and other things, for example, by separating essential and non-essential amino acids. Somewhat ironically, in this talk, thinking about the future of stable isotope analysis in Polynesia, I actually want to go back to our oldest and most well-established technique of carbon and nitrogen stable isotope analysis for dietary reconstruction. And the reason that I want to do this is because I think it's really hitting a turning point in our discipline where this method is becoming very accessible, and I would love to see this being taken on and incorporated into more of our multidisciplinary studies, especially about human-environment interaction in Polynesia. So I'm going to be discussing and hopefully encouraging you all to think a little bit more about integrating this into your own toolkits. Uh, it's become much more affordable. It's become much more accessible to do this analysis, especially because we have a really well-established and standardized methodology now. There's basically a solid recipe you can follow. You could perhaps give it to a graduate student so you don't have to do it yourself. And we also have very good post hoc quality control indicators so that you can tell whether your analysis has succeeded. If you don't want to do the lab analysis, another thing I wanted to highlight is that many radiocarbon labs will also integrate separate analysis of carbon and nitrogen stable isotopes, usually at little or even no cost. So if you're in a context where it would be appropriate to do a lot of radiocarbon dating of your faunal materials, consider integrating a stable isotope study. And finally, I hope that we can think a little bit more creatively about integrating these studies with animals. So far in the Pacific, most of the carbon and nitrogen analysis that has been done has been focused on human diet. And when animals are analyzed, they're quite often an afterthought or incorporated as just baseline potential food contributions to human diet with small and growing exceptions. And so, I was trying to think about why, as this field is growing so rapidly and we really can't consider carbon and nitrogen analysis as cutting edge anymore, why we're not really doing this as much in Polynesia as perhaps other regions, especially in terms of zooarchaeology. And I think part of that is that this subdiscipline is still emerging. The other thing is that preservation, as we know, is a constant challenge in Polynesia. You have to have the right context. You have to have zooarchaeological or other collections that are amenable to this type of analysis with good collagen preservation. And I think training opportunities have been a challenge. I think people in labs end up getting filtered to regions where there are better preservation conditions or a longer kind of body of work in this field. And I think on the other hand, that also means that broadly people in our region don't necessarily think about isotopes that much or maybe know all of the interpretive power that they can offer. So I hope I can share a little bit more of that through my past work with rats. So I think people talk to me a lot about my work and come away with, I don't know why I would care about what a rat is eating. So I hope I can show today that there's a lot more we can understand with this kind of method in terms of rats. So what we've kind of developed is this idea that, yes, you can use this to reconstruct diet, but you're also looking beyond diet when you're getting into your zooarchaeological assemblages, and you're really reconstructing entire food webs, especially if you can incorporate additional faunal materials or perhaps floral materials as well. And we're looking at how nutrients, in this case specifically carbon and especially nitrogen, are flowing through these food webs and how people and human activities are modifying this on an isotopic scale. So there's a lot of exciting potential to look in this very smaller, more refined way at how nutrients are flowing in these systems, it requires a sort of a shift. Because isotopes are not really something that can operate in a vacuum. You actually have to have a lot of prior information in order to interpret your data. What we get after our analysis is just a number, right? And then we have to bring in a lot of other lines of evidence that tell us what this number is saying. And so it's a shift of thinking about, can isotopes tell me about what kind of land uses occurred in the past? Probably not. You probably need to have a lot of prior information for isotopes to be useful, but what they can do is then tell you about what the impact of these activities that you know are happening may have had on social and environmental systems. So I won't dwell on this too much, but just to make sure we're all on the same page, the way dietary reconstruction works is this succeeds because isotopes move very predictably through food webs. So at the base of the food chain, C3 and C4 plants, due to their different photosynthetic pathways, end up with very different carbon isotope ratios with lower values for C3 plants. Most Polynesian economic plants are C3 plants. The notable exception is sugarcane as a C4 plant with higher carbon isotope ratios, and these are carried up the food chain. Carbon can also, to a degree, distinguish terrestrial versus marine resources, and this is because marine resources also resemble C4 plants in their isotopic signature. So as long as most of your terrestrial system is C3, you can also use carbon to distinguish terrestrial and marine sources. This this cartoon is meant to represent nitrogen's role in this. So nitrogen is primarily useful for determining a position within a food chain. On the left, it's a terrestrial system model, right? So there's really only one step from plants to animals. And then if someone is eating the animals, that's one more step. So there's a 3 per mil increase for each step in the food chain. Marine systems are much more complex. There are a lot longer consumer chains, and therefore marine resources tend to have much higher nitrogen than terrestrial systems. And this is how we can also distinguish different food groups. Okay, so that's the dietary aspect. And then if you have a bunch of different plants and animals, you can analyze all of them and start to reconstruct this food web. Things kind of space out in this way where you have marine resources with very high nitrogen and very high carbon. And then on the opposite side at the bottom left here, we have terrestrial C3 resources with low carbon, low nitrogen. I put pigs in 2 different spots. This is actually inspired by Barry and Mike Richards' work in the Marquesas, where this is one of the earlier studies that did focus in on animals, and they noticed that there are 2 very distinct groups of pigs, and this can speak to husbandry strategies as well. Okay, but the part that I really want to get you excited about today is the fact that nitrogen is actually much, much more complicated than this. There are a bunch of different factors that influence how nitrogen is entering into and moving through food webs, and this includes the aridity and precipitation in an environment, the forest structure, even if it's an open versus a closed canopy system, whether people are using fire, if they are manuring or otherwise working the land. All of these things actually impact nitrogen flows at the very base of food systems, in soils, and moving up through the food chain. So in a way, it's kind of bad news if you are trying to simply reconstruct a diet of a person or an animal. But in another way, it's very exciting if you can kind of shift perspectives, and especially what I've been working on is thinking about the commensal animal, right? And this environment that it inhabits, sort of at this nexus of human and natural systems. So there is a dietary component, but there's also this broader reflection of the landscape. So commensals are interesting for this because they are living in very close proximity to human settlements, but their diets generally are not being managed by people. We kind of have a broader interpretation of commensals in the Pacific, so pigs and dogs are included in that, but may be managed. So I'm more thinking about rats in this context. So again, here, because of all of these variables with nitrogen, you may end up I have a little cartoon rat that I've put by all my graphs to remind you that mostly what I'll be speaking about is rat data. So these data points are individual rats from the Kohala Field system on Hawaii Island and Kahiki Nui. And you can see, if you compare it to this entire food web, the rats are going across the whole span, but you can also see that they're clustered into two very different groups. And so the North Kohala rats are about, oh my God, no. Okay, 7 per mil higher. So environmental differences can make a huge difference in rats, and it has nothing to do with diet. It's all environment. Okay. So, and you may have seen this before. This is our study where we looked at 5 different islands, and we noticed a universal decline in nitrogen across all of these island environments. And so that was a very exciting pattern to discover, and we've argued that this is a result of human land use after arrival, that we see this kind of universal trend of declining nitrogen. Pat really set this up nicely yesterday, so I won't go into it too much, but just to say, again, on Mangareva Island, where we see that the entire system is really dependent on bird guano for nutrient inputs, we think that this dramatic decline in nitrogen is again due to the elimination of seabirds. We see rat nitrogen decline along with the elimination of seabirds, and this is probably 2 different things happening. It's an elimination of a high nitrogen level dietary input for rats, but it's also a lowering of the island baseline in general because it's no longer being fertilized from this high nitrogen content. You can see on this graph here, just plants fertilized with seabird guano versus plants that are not fertilized with seabird guano, how different their nitrogen isotope ratios are. And again, that gets carried up through the food chain independent of diet. I'm going to go real fast now. In the Marquesas, I think that we are seeing different things because on larger islands or islands that are less dependent on guano, that impact should be a little bit less severe because we have other sources of nitrogen going into the soils. So here instead, what we've argued is that this is actually a shift in land use. What we're seeing is that the blue dots are earlier, and then over time we're getting that lowering down, but we're getting a lowering not just of nitrogen, but of carbon as well. So a shift away from those high trophic level marine resources, perhaps, into a more intensified terrestrial agricultural production system. So I kind of thought this was the end of the story. You know, you could do this on every island. You could look again, and what you'll see is this declining trend of declining nitrogen. But what you'll notice if you have good enough eyes, Tukapia is here on the left. We only have a single data point on this graph for the Tuakamali phase, so that's AD 1200 to 1800, and it fits with the trend, but it's not exactly a population. So we went back and reanalyzed some of these collections. You get 2 cartoons now because we added pigs and rats. So pigs are in the gray on the left and rats are on the right. And what we found when we added more specimens from the Tuakamali phase is this trend of declining nitrogen is actually reversed. So this kind of fits with earlier interpretations by Kirch and Yen of these large-scale changes happening on Tukapia, where this system kind of moves from smaller-scale agriculture and intensifying, shifting cultivation, and then this shift sometime in the Sinapupu where burning stops entirely, and the agricultural system transforms into one of multi-story arboriculture. And so we argue that actually this change once again dramatically transforms the way that nutrients are flowing through this system. And we do argue that this is evidence of perhaps sustainability, that this shift allowed Tikopia to kind of move into a higher carrying capacity and much more long-term sustainable production. But I want to emphasize before I leave, because it would be very easy to fall into these traps, it's important to recognize that nitrogen stable isotope ratios do not quantify nitrogen itself. So just because you see a lowering in nitrogen isotope values, that does not mean necessarily that nitrogen is being taken out of the system. We were able to argue that for Mangareva because we have plenty of other lines of evidence that show this. And likewise for Tikopia, I'm saying that this is sustainability, but this is using a number of other lines of evidence to make this case that, we are seeing shifts in nitrogen, but it's more characterizing the types of nitrogen inputs and nitrogen flows rather than an overall abundance or an overall productivity measure. So that's why we say semi-quantitative. Okay, so exciting. Please use rats in your analyses. I would love to see this kind of being thought through in many more multidisciplinary collaborations and see how these trends look where on all of these other islands where we see different practices happening. I had to go quickly because I also want to take a little bit of time while I have a captive audience to say that Mara and I have finalized our agreement with UH Press. And so I also wanted to announce that we are going to have our first issue of the Journal of Polynesian Archaeology and Research. It will be coming out in the spring. This does mean that the Rapa Nui Journal and Hawaiian Archaeology have had their final issues. The Rapa Nui Journal will be coming out with its last issue sometime this year, maybe early next, we're not quite sure. And then we will be switching over to this model, and I'm happy to say we can provide entirely open access and rolling submissions, no charge for open access, all of that good stuff, because it's being supported by our respective associations, Society for Hawaiian Archaeology and Easter Island Foundation, of which many of you are members, so thank you for your support. Yeah, I think I covered it. We have a website and everything. This is not the cover, but we don't have a cover yet. So image to be developed. Okay, thank you so much.